The first presentation was devoted to the principle and approaches of design-driven bioengineering and biotechnologies towards the application in personalized and precision medicine. The second one, just logistically connected, is just the specific example what we have prepared for being used as one of the molecular tool for diagnostic monitoring and therapeutic. For autoimmune and inflammatory diseases would require greater understanding of a patient, and now for pre-illness person at risk to make the therapy personalized for either of those substances. In this sense, the identification, impact, and implementation of diagnostic, predictive, and prognostic biomarkers remain the holy grail of platforms, algorithms, and protocols which are the crucial for PPM. It's just strategy for discovering biomarkers to be used in the area of PPM guidance. So it's necessary to identify biomarkers of principally new generation and to create a fundamentally new strategy based upon not only clinical, but subclinical and predictive recognition of those biomarkers long before the disease clinically manifests. The latter is so much valuable and important since chronic autoimmune inflammation is about 40. Or is structured to consist from different stages, including subclinical and clinical ones. Subclinical and clinical illness. Meanwhile, the utility of predictive biomarkers to monitor and to manage chronic autoimmune inflammation and to predict stepwise transition is dependent on three key parameters, which must be carefully assessed: sensitivity, specificity, positive predictive, and prognostic values. Just look, why biomarkers today, biomarkers more fail to reach the clinic? Why? Biologists, technologists, engineers, and chemists fail to do it. So, accurate prediction is crucially vital for prevention of autoimmune. So, and the targeted treatment could thus be given to those individuals who are most likely to develop the disease. Among the best validated predictive biomarkers today are autoimmunity-related ones. Look, it's just a gift from my friend, the director of the Center for Autoimmune Disease from Cell Disease. So you understand me, Cell Disease. You put the shot to predict risk of the chronification and thus disabling the chronic autoimmune disease are preceded by a long subclinical symptom-free phase. MS, multiple sclerosis. Just one of the chronic autoimmune diseases resulting in the destruction of myelin by different tools, including autoantibodies. The crucial step in the pathogenesis of the disease is a primary myelin damage mediated by those autoantibodies. A spectrum of those antibodies occurring in MS patients vary a lot. Along with canonical antibodies, some of the families proved to occur are antibodies armed with catalytic antibodies or enzymes, antibodies, enzymes. We just published it 12 years ago in now. And that belongs to antibodies with a picture of functionality. It's just the illustration how their catalytic activity is working within the frame of molecular antibodies. The example of antibody protein is a source of primary catalytic antibody-proteases are IgG found in the nucleus of autoimmune disorder. To cleave targeted out the antigens that to act as informative biomarkers to monitor both clinical and subclinical causes of the disorder. Look, please. Bronchial asthma. To cleave was intestinal peptide, VIP, as the major targeted out the antigen. Targeted site. The other example is the panel of antibody-proteases detectable in autoimmune myocarditis, very severe disorder, AIM, to cleave targeted cardiac myosin. It's just the outcome. In autoimmune thyroiditis, AIT, thyroid globulin, TG, and thyroid peroxidase, two targeted antigens cleaved by antibody protein. A situation of much greater in MS, multiple sclerosis, which is the chronic autoimmune inflammation in central nervous system CNS, resulting in demyelination, action loss, and development of disability. 16 years no longer in that. We have established that MVP, briefly, myelin basic protein, MVP. Autoantibodies harvested from MS patients and mice with MS, and now in mice, MVP specific proteolytic cleavage is secured of the MVP molecule. The activity of antibody proteins is markedly dispersed between MS patients and healthy control. Also, in patients with different forms. Look. Aggressive exacerbation, regression, more white stabilization, white. And finally, the activity demonstrated also significant correlation with scales of demyelination, neurological deficiency, and thus with the disability of the patients, as it's seen from the specific clinical ADSS scores. You see, aggressive, high activity of antibody proteases, moderate, low. Of the greater value is a sequence specificity of antibody proteases to recognize specific sites within the molecule of MBP, myelin basic protein, which was analyzed by screening peptide cleavage products by a combination of reverse phase chromatography, SDS, electrophoresis, Western blot analysis, and mass spectrometry due to CELD protocol. As you might see, antibody-mediated proteolysis of MBP results in generating a set of peptides with molecular masses ranged in various but fixed boundaries to suit common principles of the molecular architecture of MBP. The final statistical data revealed 5 sites. 1, 2, 3, 4, 5. I would just faster. Most of the sites are located within 43170 region of MBP, which are just immunodominant. 2 sites from the set located within this sequence would comprise 1 to 2 more sequences that, whilst being sequence-specific, highly immunogenic, both proved to be specific at very aggressive cases. Moreover, both subsequences have been proven to be major MBP targets to be attacked by antibody proteases in patients with mostly aggressive courses. 2 extra sites from the same 43170 set are located within those subsequences, with being sequence-specific but low immunogenic and inducers of moderate cases. The strongest one is the smallest 8298 subsequence. A weaker one, longer 81103 subsequence. A site with the lowest immunogenic properties is formed by the rather long subsequence. The final step of the studies concerned the evolution of antibody-associated proteolytic activity at different stages of the disease progression. For those purposes, we have been using MS patients, their relatives, and healthy controls. You see that the activity of antibody proteases in MS patients significantly higher than at the relatives, MS relatives, but those higher than in healthy controls. So some of MS relatives might be pre-illness persons at risk. As the disease progresses to transform from subclinical to clinical stages, the activity of antibody proteases is being escalated to reach the indices we have established for MS patients. Initially, at pre-illness persons at risk in half a year, in 2 years, in 3 years, in MS patients. No time in detail. sequence-specific antibody-proteases have proved to be greatly informative and thus valuable as biomarkers to monitor MS at both clinical and subclinical cases. The primary translational potential into bioindustry and clinical practice of this knowledge as a whole is in the rational design of new therapeutics. Therapeutic antibodies is the hugest family, but no therapeutics being based on catalytic antibodies yet. Emerging have created a vast range of novel antibody-based therapeutic which specifically target biomarkers of disease, including MS. Today is the central portion of biopharma industry. So antibody protease engineering would offer the ability to enhance or alter their sequence-specific activity to expand the clinical utility of the absolutely new translational tools. Of special interest in this context are nanobodies. Globally, in this sense, catalytic antibodies, antibody proteases in particular, can be programmed or reprogrammed to suit the needs of the body metabolism, or could be designed for the development of principally new catalysts, not natural, artificial, with no natural counterparts. Of tremendous value are antibody proteases directly affecting the remodeling of tissues with multi-level architecture. For instance, myelin, which is crucial for MS progression. In summary, targeted antibody-mediated proteolysis could also apply to isolate from catalytic Ig molecules catalytic domains directed against encephalotogenic epitopes. Further studies on antibody-mediated MPP degradation and other targeted antibody-mediated proteolysis may provide biomarkers of new generations and thus a supplementary tool for assessing the disease progression and predicting disability of the patients and pre-illness persons at risk. In this sense, a year ago, I first met with two biopharma companies, one Japanese, one from US. Now it's already 7, and they are absolutely ready. We have now met with lots of competing companies. They are absolutely ready to open up to $500 million, are ready to provide for implementation of the outcome into bioindustry. For me, it's unexpectedly. Thank you so much for your patience.